Up here, an integrative systems approach to understanding cell type-specific gene regulatory networks and application to macrophages, which are very important in the processes of atherosclerosis. Stephen, welcome to you.
Can everybody hear me okay? Hey, how about in the back there? Everything okay? Great. Thanks very much for that kind introduction. I'm delighted to be here today at the University of Michigan to tell you about my research program, which is focused on using integrative computational methods to map gene regulatory networks, and on using these networks to uncover molecular transcriptional regulators of pathogenesis. The work that I'll be describing today was carried out in the labs of Ilya Smulovich and Alan Adaram at the Institute for Systems Biology and at the Seattle Biomedical Research Institute. It'll be a 5-part seminar. I'm going to start by describing at a high level the problem that my research program is addressing and its relevance to biomedicine. I'll then describe the computational approach in some detail. And then I'll present 2 applications of this computational approach, both in the area of athero. And I'll conclude with just a few slides on some other areas where I've worked and where the long-term vision for where the research program is going. Biology and biomedicine is probably well appreciated by everyone here that biology and biomedicine are being transformed by high throughput measurement technologies. This transformation is being fueled by the rapid decrease in the cost of data acquisition, which is probably illustrated most powerfully by the cost of DNA sequencing, which has dropped by an astounding 4 orders of magnitude over the past 10 years. And this is driving an explosion in the growth of high throughput measurements of such as functional genomic and epigenomic data sets in nearly every mammalian cell type, as well as fully sequenced mammalian genomes, cataloging of natural variants, and genotype to phenotype studies. Today, I hope to convey, if I accomplish one thing in my talk, I hope it is to convey my enthusiasm for integrating these types of high throughput measurements to uncover key drivers of pathogenesis. So this omics revolution has led to several different unbiased approaches to try and gain insight into the molecular basis of pathogenesis that I just want to kind of put my work in the context of some of these paradigms. The first are genome-wide association studies, which attempt to relate variant to phenotype. The second are gene expression profiling studies, which attempt to relate transcript or protein to phenotype. And the third are expression QTL studies, which attempt to relate variant to transcript. Each of these paradigms has its own inherent limitations and blind spots. And so there's been intense interest in the last 10 years in strategies to integrate findings from each of these paradigms into a combined network. However, the challenge that we're faced with is how to gain insight into causal regulators of pathogenesis. And so for that reason, my research program is focusing on gene regulation in a specific cellular context. So I'll define a gene regulatory network as a molecular interaction network that connects upstream intracellular signaling cascades to the transcription factors that they control, to the DNA cis-regulatory elements that they bind in order to regulate the expression of downstream genes and control their functions in the cell. So I'm focusing my research program on understanding gene regulation, first because we can take advantage of what remains today arguably the only truly global and unbiased quantitative cellular measurement that also has good dynamic range. Second, the transcriptome. Second, because transcription factors often control many downstream genes with related functions, and the upstream signaling cascades for transcription factors are thus of significant potential interest from a therapeutic standpoint. And most importantly for today's talk, because we can take advantage of chromatin structural measurements and epigenomic measurements as well, and in humans, expression QTL information to improve our ability to map gene regulatory networks. But for the moment, our the emphasis will be on identifying these cis-regulatory elements and the regulators that bind them in our specific cellular context. So the classic approach to this is to use cultured primary cells or cultured cell lines and to profile the transcriptional responses of these cells of interest to a variety of cellular perturbations, which could be drug treatments or environmental stresses, ideally in full time courses, and under the premise that genes that are co-expressed across a variety of time points and conditions likely have regulatory elements, upstream regulatory elements in common. These genes are clustered, and then clusters of genes that are co-expressed are mapped to the genomic regions flanking their transcription start sites. And then these genomic regions are bioinformatically analyzed to identify matches to transcription factor binding site sequence patterns that have been compiled from high throughput in vitro or cellular protein DNA binding assays. And when we find a binding site sequence pattern, such as the high mobility group box pattern shown here, whose matches are statistically overrepresented within the genomic regions flanking the start sites of this cluster of co-expressed genes, that is taken as an indication that there may be a functional association between the transcription factor that recognizes this pattern and the pattern of co-expression. So the cell type where I have been developing these computational methods, and that is a long-term interest, it's a core area of interest from an application standpoint in my lab, is the mammalian macrophage. So macrophages are an important cell type in the innate immune system. They're both have phagocytic function as well as can present antigen. They're important for both health and disease. So in health, they're vital for host defense against infection. They also help to promote wound healing, and they're important for maintaining tissue homeostasis. But they're also a bit of a double-edged sword, because macrophages can, in cases of non-resolving inflammation, macrophages can contribute to tissue damage, they can present autoantigen, and they can drive chronic inflammation. A biomedically very relevant context in which macrophages can drive chronic inflammation is in atherosclerosis. So atherosclerosis is a chronic inflammatory disease of the large and medium-sized arteries characterized by lipid-rich focal lesions called plaque, which preferentially develop at points of elevated shear stress in the vessel walls, such as at arterial branch points. Atherosclerosis tends to develop silently over a decade, and typically the first overt clinical symptoms occur when there is an acute event, such as myocardial infarction or stroke. Macrophages are present at all stages of the disease process, from the earliest fatty streaks through advanced complex lesions, and where macrophages can contribute to the thinning of the fibrous cap and lesion destabilization. And the resultant plaque rupture enables the formation of thrombus, which is what drives these acute clinical events. Heart attacks and strokes, despite the widespread use of statins and antiaggregants and ACE inhibitors in at-risk population, heart attacks and strokes are still very frequent in the United States. 1.7 million occur per year. And cardiovascular disease is thought to be responsible for approximately 1 in 4 deaths in the United States. So despite the successes of statins and other drugs that target classic risk factors for cardiovascular disease, cardiovascular disease remains epidemic in the adult population, and new therapeutic approaches are urgently needed. So there is significant interest in the macrophage as a potential cellular therapeutic target for atherosclerosis because of its role in lipoprotein metabolism and inflammation in the vessel wall. So cholesterol is carried in the blood by lipoprotein particles, for example, low-density lipoprotein, or LDL, which is sometimes referred to as the bad cholesterol. And LDL particles can become entrapped in the extracellular matrix in the vessel wall, where they are susceptible to oxidative modification, yielding an ox-LDL species, or oxidized LDL, that macrophages internalize through their scavenger receptors in a largely unregulated fashion. And when macrophage internalization of oxalate outstrips the macrophage's ability to metabolize and efflux the lipid and cholesterol, it leads to a foam cell phenotype, so named for the abundant cytosolic lipid droplets that give the macrophage a foamy appearance by light microscopy. So this foam cell formation elicits a variety of macrophage responses that are on balance pro-inflammatory and incite atherosclerosis. So I want to understand comprehensively the gene regulatory network that governs these macrophage responses to atherogenic lipoproteins. However, it must be acknowledged that studying atherosclerosis, as well as other chronic inflammatory diseases, presents significant challenges. So first, the disease is very slow to develop. So even in animal models, the experiments are very time-consuming. Second, there is significant intra and interlesion heterogeneity. Most importantly for molecular profiling of inflammatory lesions, the cell type composition of the plaque changes during the course of the disease. Third, in humans, the risk of cardiovascular disease, and in particular coronary artery disease, is thought to be controlled by complex genetic interactions, and most common risk alleles that have been identified to date have very modest effect sizes. In live humans, lesions are also relatively difficult to access, which complicates human studies. And in the most commonly used animal model, the mouse, lesions are quite small, which severely limits the amount of cellular material that can be obtained for molecular studies. So it's almost axiomatic that to gain a comprehensive understanding of the molecular basis of pathogenesis of chronic inflammatory diseases, we're going to need to use a combination of systems, high throughput measurements. For example, so I've already mentioned the importance of genotype to phenotype studies, such as GWAS studies, and I'll just touch on that at the end of the talk. In our lab, we're also using proteomics to characterize protein, transcriptional regulatory complexes that regulate cholesterol-responsive genes in macrophages, but I won't say any more about that today. And lipidomics is also an important tool to understand the changes in levels of bioactive lipids in macrophages and in the plaque. But for the moment, I want to focus on what can be learned from a system-wide standpoint using the transcriptome. And as I mentioned before, for cultured primary cells, we can profile responses to a variety of cellular treatments or perturbations, treatment with various oxidized lipoproteins, etc., and gain a fairly fine-grained clustering of genes into co-expressed groups, and thus have good statistical power for detecting statistical overrepresentation of transcription factor binding sites, shown in yellow, cyan, and magenta. But this is just a simple model of pathogenic processes in a simple in vitro cellular model. Ultimately, we want to get to a molecular understanding of the changes that occur in vivo. And here, for cells derived from inflammatory lesions, we can typically profile only a comparatively limited number of time points and conditions. So we may be able to only stratify genes just based on their ratio with lesion grade or their ratio during lesion progression versus regression. And so this is going to limit our statistical power for detecting transcription factor binding site enrichments. So we need to improve the accuracy with which we can detect transcription factor binding sites. What are the barriers to accomplishing this? Well, the first barrier is that in mammals, cis-regulatory architecture is very sparse and spread out in the genome. So while we know that the proximal promoter is important from a gene regulatory standpoint, there can be distal regulatory elements that, from a one-dimensional view of the chromosome, can be tens of kilobase upstream. But from a three-dimensional standpoint, these distal elements can directly interact, can be proximal to the transcriptional complex and have an important regulatory function. However, just bioinformatically analyzing all genomic sequence far upstream of the transcription start site to look for matches to binding site sequence patterns leads to an unacceptably high rate of false positive matches. That is to say, pattern matches that do not correspond to transcription factor binding sites and certainly not to functional regulatory elements. And just using phylogenetic conservation information which works so well in simpler eukaryotes to help distinguish bona fide regulatory elements from chance matches to binding site sequence patterns, this will not solve the problem by itself in mammals, as can be easily seen from the fact that there are transcription factor binding site locations that are not broadly conserved across placental mammals, shown here and here. The second barrier is that, as was illustrated so well by the big Nature paper from the Mouse ENCODE project about 8 months ago, regulatory elements in mammals tend to be very cell type and tissue specific. Okay, so these authors characterized 19 different, a variety of epigenetic and chromatin marks in 19 different mouse tissues, and they compiled a tissue specificity index for each of these regulatory marks. And to boil down their paper, they found a preponderance of these regulatory elements are highly tissue specific. So today, it's probably well appreciated that ChIP-seq is an essential tool for decoding the complexity of mammalian gene regulation. So I want to just define this assay. So this is chromatin immunoprecipitation with high throughput sequencing-based detection. Okay, so this is an assay that can detect the location of a DNA-binding target protein or covalently modified protein genome-wide. So if, for example, you do an IP using an antibody directed against acetylated histone 4, it would produce a numeric track along the chromosome, like something looking like this, where a peak in this track might correspond to a cluster of nucleosomes where the histone amino terminal tails are acetylated, corresponding to decondensed chromatin. So about 5 years ago, anticipating the future importance of ChIP-seq as a tool for understanding mammalian gene regulation, I developed a software pipeline for analyzing ChIP-seq data that was specifically adapted for both epigenetic as well as transcription factor ChIP-seq experiments. And as a pilot application, we used this computational pipeline to analyze data from a combination of 5 transcription factor ChIP-seq assays that we carried out in primary mouse macrophages. The goal of this pilot study was to look for hotspots where transcription factors are binding in the genome. And when we lined up this track of transcription factor binding signal alongside signal from a ChIP-seq experiment for acetylated histone 4, we noticed a correspondence between transcription factor binding hotspots and these valleys in the signal for histone acetylation ChIP-seq. So we devised a computational score representing the depth of these valleys, and we sought to use this score in a sequence-based framework for detecting transcription factor binding sites. We found that indeed, if we augment classical sequence-based detection of transcription factor binding sites, we can improve the accuracy for detecting these binding sites by 50%. So this led me to want to more broadly use epigenomic and genomic data sets to improve our insight into gene regulation. So I have systematically aggregated ChIP-seq, a variety of data sets for primary mouse macrophages, including 102 transcription factor ChIP-seq data sets, which is the buzzword for that is the cis-strome, spanning 23 different transcription factors, 63 epigenomic ChIP-seq data sets spanning 7 different epigenetic marks. These are marks that include actively transcribed regions, facultative heterochromatin, constitutive heterochromatin, and so forth. I carried out a DNA-seq assay in my lab to map regions of open chromatin, and we also obtained DNA-seq and FAIR-seq data sets, which also give insight into open chromatin from the ENCODE project. We also obtained from the Phantom project experimentally determined transcription start sites from deep cage assays, and we also carried out POL2 ChIP-seq assays in our lab. And finally, in humans, we can use expression QTLs. So for example, there have been multiple studies of expression QTLs in monocytes derived from segregating populations for lipid levels in humans. So I want to point out that for these mouse macrophage data sets, about 80% of them came from public sources and only 20% came from our lab. So I think this speaks to the potential utility of these approaches in other cell types and situations where we can leverage these data sets from the Mouse and Code project and similar data resources. So I devised a computational strategy to integrate these various data sets, and it's a two-part strategy that I call regulatory element machine learning to infer networks instructing specific cellular expression. And it's a two-part approach. The first is to use a supervised learning algorithm to combine all of these cellular epigenomic and chromatin structural measurements. And in humans also the idea would be to combine expression QTLs as well, to predict the locations of probable cis-regulatory elements genome-wide. And then the second part of the approach is for genes that are transcriptionally dysregulated in our cellular response of interest, for example, responsive macrophages to atherogenic lipoproteins, within these regions, we carry out a statistical test for overrepresentation of sequence matches to transcription factor binding site patterns. And from that, as I'll show, we can bootstrap a regulatory network. So we're focusing on a cell type specific gene regulation. It's an integrative method that tries to leverage epigenomic as well as chromatin structural information. And we're using a model-based approach to identify key regulators and their targets. So how does it work? We're binning the chromosome into 100 base pair intervals. Okay, and then within each interval, we're taking advantage of all the sort of cell type generic information that we have access to from annotation of the relevant genome, for example, the annotated mouse genome. So we have information about, we have scores representing phylogenetic conservation across placental mammals. We have GC percentage information, proximity to the nearest CPG island, and so forth. So these are all arguably potentially relevant to predicting whether you're in a regulatory region. But very importantly, we're leveraging all of these cell type specific measurements that I've discussed before. Now, anytime you're talking about supervised learning, you need to have a training data set. That's fundamental to the whole enterprise. And so here, what I did was to combine those 102 transcription factor ChIP-seq data sets. So they span 25 transcription factors and their binding locations are highly correlated in the genome. So we're gaining insight into these hotspots where transcription factors bind, and using those locations as a set of true positives to train this supervised learning algorithm. Okay? And then we can use it to map probable cis-regulatory regions genome-wide. So then for genes that are responsive in our cell type of interest and our cellular perturbation of interest, we map to, again, the large genomic regions flanking the transcription start sites. But here we use that supervised learning algorithm that I mentioned to map probable regulatory regions. And then we bioinformatically analyze the sequence within these regions to look for transcription factor binding site sequence pattern matches. When we find one, we can map to its putative target genes. And if we do this for all of the transcription factor binding site sequence patterns, we can gain a compendium of cis-regulatory elements that are potentially functionally associated with our cellular response. We can map to their downstream targets, their putative downstream targets, as well as their transcription factors that recognize those cis-regulatory elements and their upstream signaling cascades through molecular interaction databases. So this is kind of how the data look when you organize them along the chromosome. So I just want to point out here that, importantly, in a given 100 base pair interval, which is about this distance, we're not just taking into account whether you overlap a given epigenomic feature, but we're also taking into account proximity to the nearest feature of each type. Okay? So you may not directly overlap a histone acetylated region, but you may be directly proximal to one. And so that's taken into account in the learning algorithm. So computer science has provided us with a wealth of supervised learning algorithms to choose from. And so I tried 3 of them in this pilot study. to integrate these largely continuous valued features to try and predict the locations of these transcription factor binding hotspots genome-wide. And the three that I tested were adaptive boost, the support vector machine, and a random forest model. And in terms of prediction performance measured by the curve, the curve representing the trade-off between sensitivity and type 1 error. So the closer the curve gets to the upper left corner, the better a job the model is doing. The random forest was the highest performing, and it was also by far the fastest. Okay, so this was pretty much a no-brainer. I'm using random forest, and it is an ensemble classifier that can take advantage of both continuous as well as categorical features. Categorical means discrete. And you're basically training an ensemble of decision trees. And each decision tree is based on a different subset of the features. And in a given 100 base pair interval, then, each decision tree votes on where that interval does or does not coincide with a regulatory element. And so the fraction of trees that vote yes has a nice probabilistic interpretation, which is very convenient for incorporating into statistical network inference downstream. So this is how the voting fraction might look if you thresholded it at 1/2 along the chromosome. And not surprised. Okay, so the salmon-colored boxes represent cell type generic information. So CPG, island, proximity, GC content, phylogenetic conservation, and the like. And the baby blue blocks represent the cell type specific information that we've integrated into this supervised learning algorithm. And then the voting fraction scores in each 100 base pair interval, thresholded at 1/2, which is why you don't see anything below half here, are displayed along here. So naturally, it's only natural. I think I was curious. So that's just one of the features that I incorporate. So this is, it occurred to me that your distance along the chromosome might potentially be connected, because if you're far out towards the telomere versus internal to the chromosome might be relevant, because in terms of correlation with heterochromatin, that it occurred to me that might be an informative feature for the machine learning algorithm. So that's just taking the whole chromosome and then scaling it between 0 and 1. So it's just a nice. So that's not something that I tested. So the normalized distance along the chromosome, I don't believe it didn't turn out to be a particularly strong feature, but the actual physical distance in megabase along the chromosome, I didn't include. But because of the significant differences in absolute lengths of the chromosomes, my worry was that a decision tree you trained on data from one set of chromosomes might not generalize to another as well. But I haven't tested it, so. Okay, so to boil this down, so it's only natural to want to ask, well, what subsets of features were actually useful, right? So we've combined 19 features, and so again, the closer the curve gets to the upper left corner, the better job those combination of features do. And so all 19 features did the best, and a random model, which this is literally randomly guessing whether at any given bin coincides with a binding hotspot, of course, does very poorly, and you see, a little bit of an improvement if you take into account proximity to the transcription start site, but it's not until you get into including cell type specific measurements shown in purple and blue that you're really getting a significant bump in performance. So that's also more clearly illustrated if you use a measure of feature importance called the Gini index. So the longer the bar is, the more important that feature is in discriminating between the true positive and true negative. Excuse me. And so, first of all, I want to show you that the preponderance of blue bars up top is, showing that the cell type specific information really is essential to the performance of the classifier, with the exception of the chromosome that you're on actually turned out to be important. So that was kind of curious, and we can talk about that after the talk if there's interest. So then the next thing that one notices is that the hashed bars, which are the histone acetylation, also were all clustered towards the top. So this is consistent with our previous work establishing that histone acetylation derived features are particularly beneficial in mapping transcription factor binding sites. So, and then I just want to emphasize that we're not just predicting that you should look only within 500 base pairs of the transcription start site. That would be sort of disappointing, since the whole goal was to do a better job by being able to go further out. And so indeed, close to 20% of the predicted regulatory elements are more than 2 kb from the transcription start site. And in the interest of time, I'll just say that we mask low complexity repeats. We scan for motif matches within all non-coding sequence in these predicted regions. And for this pilot study, I used a tandem statistical test, but to test for overrepresentation of binding site sequence pattern matches. But where I want this to go is a Bayesian approach to identify statistically overrepresented binding site sequence patterns. And so we can talk about that perhaps after the talk if there's a lot of interest. The idea is to take into account the voting fraction from the supervised learning algorithm without having to threshold it. So I don't need to apply, I wouldn't need to apply a hard threshold. And also to take into account uncertainty in the sequence pattern for the transcription factor. Probably well appreciated by many of you that these transcription factor binding site sequence patterns that have been compiled differ widely in how their degree of uncertainty in their composition. Yes. Oh, you mean in the list of, sorry. Let me back up a step. So the supervised learning algorithm is not using transcription factor binding site, but in the statistical test for overrepresentation, yes, absolutely. That's a really important distinction. And we can talk about why I have some ideas about why it's best to split them up that way, but it's a very important sort of separation of information in the method. So I have several ideas about where I would like this to go, but most notably incorporating 3D chromatin information when the resolution gets to where I would like it to be. If the resolution was sub-kilobase, I can sort of directly envision how I would pull that information into this computational approach and foresee how it would directly benefit the framework. At 40 kb resolution, I'd have to spend some time thinking about how to do it. Okay, so this now I want to very briefly talk about two applications. So I've presented the computational approach and it described how it integrates all of these various measurements. So now I would like to very briefly talk about an application of this approach to understand the transcriptional changes in macrophages that have been treated with atherogenic lipoproteins. So this work was done in collaboration with my colleague in the Aderum lab, who is also a cardiologist at Virginia Mason Medical Center, Liz Gold. So my initial goal was to uncover the transcriptional changes in macrophages in response to atherogenic lipoproteins. So I isolated peritoneal macrophages from black 6 mice, incubated them with ox-LDL, with a high concentration of ox-LDL, and they very clearly become bone cells, as can be seen by staining with the lysochrome dye oil red O, as well as with the lipophilic fluorophore, Bodipi 493503. And based on the literature, there were some transcription factors that I expected to see that would be, whose binding sites would be overrepresented for the genes that are up or down in response to ox-LDL. So I had a list of ones that I expected to see coming out of the method. But my hypothesis was that using this chromatin-guided approach, we would see a lot more. But first, we needed to do the transcriptome profiling. And so I carried out exon array transcriptome profiling of these peritoneal macrophages. And I did some qPCR just to validate that the hybridization-based measurements are faithfully recapitulating the transcript levels as they would be, taking qPCR as a gold standard, and they are. So then I used this chromatin-guided transcription factor binding site analysis method that I described to see what transcription factors binding site sequence patterns I could detect. And you always want to have a control, right? So as a method, as a sort of control method, I used a sort of naive or classic approach. Just look at all non-coding sequence within 1 kilobase of the start site and or within 5 kilobase of the start site. Okay, so that's kind of the control. And the bar length represents the enrichment ratio in the number of binding sites versus what would be expected by chance for a randomly selected group of genes that are expressed in peritoneal macrophages. Okay? So that's why we're not trying to, the intention is not to detect a transcription factor that is just associated with a macrophage being a macrophage, but instead to detect transcription factors that are associated with a group of genes that are up or down in response to OX-LDL. And so the white bars are the ones detected using the chromatin-guided approach, and it's easy to see we detected a lot more transcription factors, and in every case, we have a stronger enrichment ratio if we use the chromatin-guided approach. And that was also the case for genes that were down in response to OX-LDL. And the transcription factors that we expected to see based on the literature we detected, so that was also gratifying. And another transcription factor family, this HMG box binding transcription factor family also caught our attention for reasons that I'll get to later in the talk. So I just want to mention this TCF-LEF transcription factor family. So the transcription factors that bind these regulatory element patterns are highly connected in the protein interactome, not surprisingly, many of them complex together. And one of the transcription factors that's highly connected in this network and caught our attention because we had done some previous work on it is the stress-responsive transcription factor ATF3. And it is itself moderately transcriptionally induced in response to OX-LDL. We also have known from previous work in our lab that ATF3 negatively regulates expression of TLR-inducible, that is to say, toll-like receptor-inducible pro-inflammatory cytokines. So toll-like receptors are a conserved superfamily of membrane-bound receptors that detect microbial molecular components. So they're a part of the microbial sensing machinery that macrophages and other cells possess. And so ATF3 is an obligate repressor that, when in a TLR-stimulated macrophage, will help tamp down expression of pro-inflammatory cytokines. So we wondered whether it also plays a role in regulating macrophage foam cell formation because of the previous results that I showed you. And so I isolated macrophages from wild-type and ATF3 deficient mice and incubated them with OX-LDL. And indeed, at every concentration of OX-LDL that we tested, there was a significantly greater amount of neutral lipids and lipid droplets in the knockout macrophages compared to wild-type control cells. So we also validated this using an enzymatic assay for cholesterol esters that I worked out in the lab. And we also validated this finding by mass spec-based lipidomics for neutral lipids. So these cells, there seems to be increased foam cell formation when ATF3 is knocked out. We wondered whether that was also the case in vivo. And so we used the apolipoprotein E deficient mouse as a model of diet-induced hypercholesterolemia. And indeed, the double knockout, peritoneal macrophages from the double knockout mouse also have more lipid droplets than the age and sex-matched control animals. So ATF3 seems to be a negative regulator of foam cell formation. So we wondered about its effect on atherosclerosis. So we again turned to the ApoE knockout mouse, which in addition to being a model of diet-induced hypercholesterolemia is also a model of atherosclerosis in the mouse. And so we crossed the ApoE knockout mouse with the ATF3 knockout mouse. And we weaned age and sex-matched cohorts of single knockout and double knockout animals onto high-fat diet for 8 weeks. And at this early stage of the disease process, we found an increased lesion area at the aortic sinus in the double knockout as compared to the age and sex-matched control animals. So at least at this early stage of the disease process, loss of ATF3 exacerbated atherosclerosis at the aortic root. And using systems approaches, which I don't have much time to describe today, we think we have a pretty good handle on the mechanism. So using transcriptome profiling, I should say that we tried all of the obvious explanations, looking at plasma lipids, serum cytokine levels, and found nothing that could trivially explain this phenotype. And so that is why we turned to transcriptome profiling in mouse macrophages to try and get a handle on the mechanism. And we found that the most strongly upregulated transcript in the knockout versus wild-type macrophages encodes the enzyme cholesterol 25 hydroxylase. So, and this is an enzyme whose substrate is cholesterol, and that converts cholesterol into 25-hydroxycholesterol. 25-hydroxycholesterol, based on work going back to Brown and Goldstein in the 70s and 80s, is known to promote cholesterol esterification. This is what led us to suspect that it might play a role in our atherosclerosis phenotype. And in the interest of time, I just want to skip forward. So we think we understand the mechanism by which this enzyme, this cholesterol 25 hydroxylase gene, is dysregulated in the knockout. It's due to ATF3 directly binding the promoter of CH25H and changing histone acetylation in response to cell stress, such as OXLDL. And in order to measure the level of this oxysterol, I worked out the chromatography to detect 25-hydroxycholesterol in lipid extracts from macrophages, macrophage-conditioned media, as well as from mouse aortas. And in every case, we detected that loss of ATF3 leads to a significantly increased level of this oxysterol. And we found that if you incubate macrophages, just wild-type macrophages, if you put 25-hydroxycholesterol into the growth medium, it also promotes cholesterol ester formation and lipid droplet formation. So 25-hydroxycholesterol seems to promote foam cell formation. And we did the reciprocal experiment using the CH25H knockout. So we think 25-hydroxycholesterol is a driver of lipid droplet formation. And we're doing the ongoing studies to determine definitively whether 25-hydroxycholesterol is in fact the mediator of the atherosclerosis phenotype that we have detected in this ATF3 knockout animal. So I have just a minute or so to mention that we're also working on another systems biology collaboration that uses these computational methods with Ed Fisher at NYU and also with Liz Gold. And here the goal is to understand the transcriptional changes that occur in macrophages in regressing atherosclerotic lesions. Okay, so unlike in humans, in mouse there are various manipulations that can readily trigger regression of advanced atherosclerotic plaque. And in every case, in all the models, the commonality is that macrophages emigrate from the plaque. So we want to understand from a gene regulatory standpoint what the triggers might be that cause this egress from the plaque. So we have 2 different models that we're studying, and I don't have time to go into all of the details today, but I just want to emphasize that using our chromatin-guided analysis for transcription factors, we detected this TCF-left transcription factor as overrepresented among genes that are upregulated in plaque macrophages in regressing versus progressing lesions. So why is that important? Well, this transcription factor family is downstream of the Wnt beta-catenin pathway, okay, which is well known to control, among other things, cell motility. And so this led us to hypothesize that beta-catenin levels might be upregulated in macrophages in the plaque in regressing lesions. So I just want to jump ahead to that to show that we did a pilot study with this was a postdoc in the Fisher lab. Yulia Van Grenyuk did the immunohistochemistry. We found that there is, at least in this pilot experiment, we found increased levels of beta-catenin in macrophages in the plaque in regressing versus progressing animals. And so at least in this pilot experiment, in this one model of plaque regression, we're seeing a nice validation of a prediction that came out of the integrated computational analysis. And so now we're going to look at Wnt inhibitors, and you can imagine what might come next for that. So now I'm going to skip ahead because we're running out of time, and I want to tell you about where I think the research program is going. So this is the last slide before the acknowledgement. So the long-term vision for where I've talked a lot about mapping gene regulatory networks and some of the experimental work to try and understand the function of transcription factors in these networks. But from my standpoint, the network is really not the end goal, but rather a means to gain a better understanding to identify candidate drivers of pathogenesis and candidate disease genes or biomarkers. So the long-term vision for where I hope to take my research program is a second emphasis that complements the part of the program that emphasizes mapping regulatory networks. The second part of the program would be emphasized on using these, would be focused on using these networks to identify candidate disease genes. Most importantly, taking into account what we know from genetic association studies and the full protein interactome and so forth. And so hopefully I'll get a chance to talk with some of you about that at length. So I want to thank my collaborators, especially the Fisher Lab, and my mentors, Alan Adair and Ilya Smilovich. I also want to thank Liz Gold, who's been my long-term collaborator on this atherosclerosis work, and our technician, Irina Podolsky. And finally, I want to thank my funding source, in particular the NHLBI. Thank you very much for your time.
We have a lot of data publicly available, and you generated 20% of it. So if you were going to, is there a hierarchy of what data is actually most useful? Because a guy like me who can't generate a lot of this data de novo, can I get most of this information just by pulling it off from publicly available sources?
So I'm happy to bravely speculate based on what I've seen from macrophages to other contexts, but with the caveat that what I'm about to say is based on my observations from measurements in mouse macrophages. But histone acetylation is clearly the one measurement I would start with. DNA's hypersensitive sites also are important. If I had to make two sets of chromatin and epigenomic measurements, those would be the ones that I would start with. Which is not to say that others won't prove to be important when they become, when coverage levels improve and when we get, some of these data sets get deeper. But that would be my starting point if I were to take this into a cell type or context where I had not used it before.
When you had those bar graphs comparing your methods with that OX-LDL enriched up and down regulated genes, lots of those things were just blanks in the sense that you got, the blank bar for your method and nothing in the other method. Is that because the other methods aren't searching for it, or just because they're, completely ineffective compared to what you were doing? I mean,
So mostly it's because in the other methods we were below the statistical significance threshold for detecting an overrepresentation of the transcription factor those transcription factor binding sites. So now ultimately the real proof is, can you take the real proof of the discovery potential of the method is to take transcription factors that come out of the method and validate them in vivo, as we've done, right? I mean, that's the proof that convinces me and has me excited. But that's a long-term process. The ATF3 study was a 4-year study. So,
but actually, I was thinking a little bit also in terms of methodology there. I mean, just in terms of that graph, whether there's a a fight against a straw man there, in the sense that it is a certain technique not expected to register anything anyway. In other words, you never expected them to show up on certain of those things that you were measuring.
Well, if you're talking about the comparison to the scanning all non-coding sequence within 1 kb or 5 kb of the transcription start site, so I mean, let's say transcription factor X is indeed, its binding sites are indeed overrepresented in all of the real regulatory, all the bona fide regulatory elements near this group of genes. So the question is whether we can detect it. So you're going to see the sequence matches for those, all of those sites if you use your 5 kb plus or minus 5 kb method. The problem is you're going to see so many other false positives that it's kind of just washed out, right? Because false positives contribute to the numerator and the denominator. So, I don't know. I mean, I felt it was important to compare to something. There are other possible strategies I think that could be useful for complementary validation methods. So, for example, applying it in a simpler model system where we have comprehensive information about from deletion libraries and so forth. So, that's certainly something that would have merit as a complementary approach to validate the method. Obviously in mouse we can't do that. Sure. I think it might be best to jump out of Keynote here. And then, okay, well, now it is, it's buffered all the keyboard input and now, okay, sorry, 1 second. Back one?
So, first of all, I'm very interested to know if you predicted in advance what places you should be explain. I didn't understand what you mean by the topmost curve that
Now, if you zoom in enough, and I don't know that I can, I don't know that I can do that on this, but if you zoom in enough, it's, no. Random forest definitely outperforms adaptive boost. In fact, I did a statistical test, and there's definitely, this is the area under that blue curve, yeah? And this purple bar is the area under the purple curve, and the blue curve clearly has greater area, which it can only do if it is higher than the purple curve. Yeah.
It's a little lower.
Yeah, we'd have to. Oh yeah, yeah. It's about 5 times faster than adaptive boost. Now again with. And yes, and the performance was a little bit better. Whether that, but yes, it's significantly faster for to train. Yeah. In terms of correlating the predictions of the machine learning algorithm with cis-EQTLs and that kind of thing, or? Yeah. 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 So I remain very sanguine about the utility of EQTLs in the long run for this kind of enterprise, but it's more of something that will bear fruit in human than so that there have been 2 studies of human monocyte EQTLs, that have generated, I think, 10,000 and 20,000 cis-EQTL variants respectively. And I did map those variant locations over to mouse using liftover just to see if those locations as a feature would have any benefit in the prediction. But if they did not seem to have any benefit. Having said that, it's another story whether they would be beneficial if I redid the whole analysis in human. So that's something that's worth exploring, and because we definitely see connections between some of the transcriptional some of the differentially expressed genes that we're seeing and GWAS variants for coronary artery disease. So I didn't have time to show that, but one of the most strongly upregulated transcripts in regressing versus in macrophages in regressing versus progressing plaque is with the 3' end of that transcript is within 40 base pairs of a variant that has a GWAS, statistically significant GWAS association with LDL levels and risk of coronary artery disease. And that association has been replicated in targeted studies, in independent targeted studies. And it's also quite interesting in that it regulates, that's PSRC1, yeah, proline serine-rich coil 1, yeah. And so it's very interesting. I mean, we're definitely looking into it. So it's thought to play a role in regulating microtubule polymerization, and so one could imagine that might be connected with macrophage motility. But and on the other hand, my enthusiasm is tempered by the fact that the effect size of that allele is, about 1.1 for risk of coronary artery disease. So, I mean, like all the other common variants, it's a small effect size. So.
Yeah, I don't really know anything about atherosclerosis formation or progression other than the slide you showed there, which was very interesting. But it puzzles me, apropos what you were saying about plaque regression in the mouse model, and you said that seems to be associated with migration away of the macrophages that were stuck under the plaque shell, or whatever you would call it. And yet in your picture, at least I guess for a human model, you seem to say at the end stage that breaking of the plaque shell released this necrotic residue stuck under the plaque, and that was actually the cause of the thrombus and the serious catastrophic events. Mm-hmm. So how is the mouse letting this stuff out from under the under the shell without causing a thrombus or whatever?
So we don't know whether macrophages are egressing through the lumen or the adventitia. That is not, I mean, it's controversial, and so I'm not going to take a position on that. So the macrophages that have egressed from the plaque have been detected in lymph nodes. So we think we understand, at least in one model of regression, where those macrophages are ending up. But the specific route that they're taking is not entirely clear. And also, I want to point out that the mouse is not is not thought to be a very good model at all of lesion rupture, of plaque rupture. So the lesions that tend to form in these classic atherosusceptible mouse strains tend to be very stable lesions, and they even when they're very, very advanced, they don't tend to rupture. So the rabbit is a good model of diet-induced hypercholesterolemia and atherosclerosis, like the Watanabe rabbit. I think to get into plaque rupture, it's pigs, but then you're talking real money to do those experiments. But
Any final question? Thank you.
Thank you very much for your time.